Good morning, everybody. I'm Giovanni Barbara from the University of Bologna, and it gives me a great pleasure to welcome you in Prague for this 11th edition of Gut Microbiota for Health. And on behalf of the Board of Directors and the Scientific Committee of Gut Microbiota for Health, I think that we should be proud for this meeting because, as you can see, this room is full, and we are very happy to see many people coming to Prague, and still this meeting attracts a lot of interest and a lot of people from abroad. Today I would like to thank all the people that came here to Prague, particularly those who travel from far, and we should look at the future of these initiatives with enthusiasm, because we just ended last year the celebration of the 10th anniversary of Gut Microbiota for Health, and I think that the future is bright, and the interest around the field of microbiota is still growing, so we look forward to the next 10 years to come with a lot of enthusiasm. So with this, I would like to introduce the President of the European Society of Neurogastroenterology and Motility, Professor Kazim Aziz, who will give you a welcome here in Prague. Thank you very much for coming.
Thank you, Giovanni. And as Giovanni said, great welcome to all of you to Prague to attend this fantastic meeting. This is now the 11th edition, and we've had 10 very, very exciting meetings, and I have no doubt that this will be, again, another very exciting meeting. We have this long-standing collaboration with the AGA and with the American Neurogastroenterology and Motility Society, and together we have put together this program, which I'm sure you will find very exciting, and you have 2 days ahead of you of cutting-edge science, which I hope that you will enjoy, along with enjoying this fantastic city of Prague. Microbiome science is at the cutting edge of medical discovery, and it's really exciting to be involved in this area of research. And over the next couple of days, you're going to see and hear some of the latest research in the field. I'm very excited about it. I always think a good meeting is one where you go back with 1 or 2 really good research ideas for your own research. And I have no doubt that this will be the case at this meeting as well. And again, I'd like to thank all the organizers who've been involved in getting us up to this stage. And with those words, I will actually hand over to Gail Hecht, who's actually the AGA representative at this meeting. Thank you. Gail.
Good morning. Yes, I'm Gail Hecht. I'm here to welcome you on behalf of the American Gastroenterological Association. This has been really a very successful endeavor, partnering with AGA in the US and the European societies. I think when this kicked off 10 plus years ago, all of us were a little uncertain, where this would end up, but it's a very established meeting now. People look forward to it year after year. And to that end, I'll mention briefly that next year the meeting will be back in the US in Washington, DC, and probably the following year as well, trying to get back on a schedule that's more suiting for the European side and conflicting meetings. But let's enjoy the program together. It's very robust, great job. I want to thank the organizing committee for putting this together, and all of the speakers and audience for coming to make this a success. Thank you.
Oh, good morning, everyone. My name is Ari Sokol. I'm a gastroenterologist in Paris, and I'm also co-chair of the scientific committee. And on behalf of this scientific committee, I'm very happy to welcome you in Prague. I want also to thank our supporter for this meeting, without whom this meeting would not be possible. So particularly Danone, Sanofi, Biocodex, Bromatech, and Allergosan. So really thank you very much for that, for your support. We built, I think, a very nice program focusing on the next generation of microbiota-based therapy and nutrition with outstanding speakers from everywhere in the world. So I hope you will enjoy it, and I wish you a very nice summit. Sorry, I forgot one very important thing is to introduce the first speaker. So Jin Cheng from Chicago, who will give us a keynote lecture on the bright future of next generation microbiome-based therapy. Thank you, Jin.
Thank you, Harry. It's a great honor to be here, and I'm delighted to speak to such an august audience. So Harry asked me to talk about the bright future of microbiome-based therapies, and as I thought about it, I certainly believe there is a bright future, but I think we have a ways to go, so this talk is going to be a little bit about caution as well. To begin with, I have no disclosures to make that are relevant to this presentation. So I think that this is basically a review. There are many types of microbiome-based therapies. Some include live microbes, such as probiotics and live biotherapeutics, but they also include things like prebiotics and postbiotics, and even changing the gut ecosystem in a way that gives better fitness and advantage for commensal type of organisms. So with that said, we're going to focus mostly on live organisms, and this includes probiotics and also live biotherapeutic products. So if you look in the left-hand column, probiotics are basically microbes that have been developed for the maintenance of health. Now, originally, there were many claims that they might have some indications for specific diseases, but I think that most people in the area of probiotics have really backed off from that because these claims have been largely unsubstantiated. Nevertheless, I think that probiotics are very much part of our culture. They are either sold as dietary or nutraceuticals, and live biotherapeutic products, on the other hand, are the new kid on the block. These are microbes that have been developed for specific medical indications. They're looked at more as a drug than a nutraceutical, and they have been showing some initial very encouraging results. So just to define a live biotherapeutic product, well, let's see what happened here. These are live organisms such as bacteria. They're applicable to the prevention, treatment, or cure of disease or conditions of human beings, and they're not vaccines. So, and this is probably not new to most of you in the audience. So there are limitations and problems with probiotics. First of all, there's very little scientific basis or evidence that really support their efficacy, certainly as indicated for certain diseases. So they're not really under the purview of the FDA. There's a fairly low bar there for direct-to-consumer route, primarily based on not causing harm, but not necessarily showing efficacy. They're generally ineffective for disease indications, and there's this issue of whether they actually engraft. Now, I know that there will be some subsequent discussions at this meeting about the importance of engraftment. I personally think engraftment is important. There are really few industry standards, and quality control is very non-standardized. Medical claims are generally not permitted, and I think that over the past several 2 to 3 decades, the public has really embraced this as being important for their health. And that's really something that I think still remains to be proven. So that's not to say that probiotics don't have health benefit. So this was a study that we reported in 2004 that showed that probiotics, at least they make these small molecules that are found in conditioned media that can inhibit NF-kappa B and induce cytoprotective heat shock proteins in colonic epithelial cells. And this occurs through proteasomal inhibition. In the left panel, you see that, in the left panel, you can see that in cells that are stimulated with TNF-alpha, with a conditioned media from VSL number 3, there is a fairly rapid decline of I-kappa B, which is inhibitory, keeps NF-kappa B in check. But in the presence of this conditioned media, you can see that I-kappa B degradation is inhibited. And this is one of the mechanisms by which conditioned media from these microbes can be anti-inflammatory. On the right, you can see by the Western blot that both HSP25 and HSP72, or now known as HSP70, these are inducible cytoprotective proteins, and they're induced in a very time-dependent way with this conditioned media. Now, the other issue is that it may be a matter of timing. And this is a study that was reported by a very talented postdoc of mine a few years ago, where he looked at the impact of a dysbiosis that was created in maternal pregnant dams, and then was vertically transmitted to the pups. These were all on IL-10 knockout background. That is, these mice are very prone to the development of experimental colitis. And what happens is that in the upper panel, you can see that this dysbiosis that's inherited actually leads to the development, substantial incidence of experimental colitis. And what we noticed at that time was that the dysbiosis was missing many of these very important commensal microbes, in particular one that was 3 log units greater than the next Bacteroidetes. So we cultivated that organism and we tried to give it back late in life. And it turned out that that really did nothing in terms of changing the incidence and prevalence of experimental colitis. In contrast, while we gave that same foundational strain back early in life, this was during the pre-weaning period, during this period of immunological development, it engrafted very well. And the consequence was there was a substantial reduction of later life development of experimental colitis. So the bottom line here is that it's really important to know that the timing of interventions is very important, and many of the risks that we think may be involved with things like complex immune disorders or metabolic diseases may begin in early life. So in thinking about probiotics and live biotherapeutic products, you know, so often what we do to develop these taxa or these consortia is that we look at in vitro data. And the problem is that when you put these microbes in an in vivo setting, they act very differently, and they're almost uncontrollable. It's like herding cats. So I wanted to point out, before I do that, I wanted to mention a little bit about fecal microbial transplant, because I think that there's going to be a lot of discussion today and tomorrow about FMTs. The notable features are that you're now putting in a healthy community that's already been self-selected, and the individual components work reasonably well together. It's been shown to be very effective for the prevention of recurrent C. diff and generally is well tolerated. It's now being studied for diseases such as inflammatory bowel diseases, irritable bowel syndrome, metabolic disorders, and neurologic diseases. So delivery can be done in many ways. It's taking a donor microbiota and either coming above or going below and administering it. Now, there are limitations. First of all, most FMTs that are currently used today are undefined. That is, we don't know what's in them. And they're likely variable depending on who the donor is. And there's insufficient evidence on both long and short-term consequences. Engraftment sustainability is really uncertain, but it can be improved with pre-FMT antibiotics. And then there's really the absence of objective metrics that allow us to determine whether or not certain microbiota are healthy. That being said, you know, the FDA recently approved the first microbiome-based drug. This is called RBX 2660. This consortia was found to be effective in treating recurrent C. diff. But it should be said that the effect size was very small, but it met the sort of pre-study endpoints that were set. They found that they could prevent the recurrence of C. diff in about 70% of their subjects, and that's compared to a 58% rate just with placebo. So it's very small, but it was sufficient enough that the FDA decided to approve it. So in going forward, I think there are many limitations and challenges that we have to address. And first of all, I think it's really critical that we find some way of defining what a healthy microbiome is. And currently, there aren't any measures. I think things that we've used in the past, like diversity, 16S profiles, are inadequate and very variable among a healthy population. Without defining a healthy microbiome, I think it's really difficult, impossible to determine what an unhealthy gut microbiome, something that many of us use the term dysbiosis for. The other thing is that when we find a dysbiosis, do we know that that is a cause, or is it an epiphenomenon, or merely a consequence of disease? And that's very important because if we're going to try to correct that, hopefully it's not just a consequence, that is, we're just treating the symptom. And how should LBPs be developed and formulated? Well, this is really, again, quite variable, often just sort of cherry-picking various taxa that are thought to be healthy and putting them together in a consortium. Is there any precision with LBPs? I don't have an answer for that, but I think that's something we have to think about. And what metrics and endpoints should be used to assess the efficacy of LBPs? Again, I have no answer for that at the moment. And is engraftment important? So currently, if you look at the metrics that we use to assess the efficacy of LBPs, in Table 1, we use a variety of sort of soft clinical measures, and they basically are looking at whether or not there's symptomatic improvement. In Table 2, there are biomarkers that are fairly disease-specific that people have used, but they too are somewhat very unreliable surrogate markers. And so it's very unsatisfying at the moment in our ability to be able to assess the efficacy of these compounds. Now, I want to point out a study by Balfour's group at University of North Carolina, which I actually kind of like. So Balfour and his group went about and asked the question, well, can we put together a consortia of microbiota based off of what functional properties we think are required, in this case, in an inflammatory model, or IBD. And they started off with the IL-10 knockout mouse, and they identified this group of pathobionts called EER, which included E. coli, E. faecalis, and Ruminococcus navis. And that's shown above. So when you put this into a germ-free mouse, you see that they engraft, and it's stable for at least 5 weeks. And the cool thing is that their initial consortia that they studied, called GUT-103, was effective in both prevention and treatment. And treatment is shown in the middle set of graphs. You can see that after 2 weeks, there was the introduction of this GUT-103 that then led to a decrease in the taxa representation of EER and a replacement of, with many healthy microbes. It also worked in prevention. So if you start with that consortia and then you introduce the EER, they were able to show that not only was there a sustained protection against the EER, but also reduction of the development of colitis. Later, they developed a human-based consortia called GUT-108, and you can see on the right that it led to histologic improvement as well as increases in very important molecules such as short-chain fatty acids. So even then, I think that animal models like notobiotic technologies, while they will give us certain answers, they have limitations. And the major limitation is that you have a skewed population, particularly if you put human strains into a notobiotic mouse. And this is shown by not only a postdoc from my lab, Dr. Nafé, but also this was originally reported by Jeff Gordon's group many years ago. And that is that we as humans are very specific about the kind of microbes we take. Generally, microbes that come from non-human sources will not engraft very well. And this is just shown when you put a mouse microbiota or a human microbiota, you can see that there's significant decrease in both engraftment and evenness. The latter means that there are certain populations that are very dominant and others are not, and these are the ones that have engrafted. So I want to end with this concept of seed and soil. And this is really based off of the very important need to understand ecological principles. So the gut is basically a number of ecosystems that are connected in series. Each has a very different type of microbiota. Generally, those that are in the small intestine, for example, are facultative and oxygen tolerant, whereas those that are in the colon are fairly strict anaerobes. So one of the problems is that if we're taking from the FMT, mostly anaerobes, the question is, are we actually replacing or repairing things that are in the small bowel? That's unknown. The other thing is that, industry and academics has mostly focused on the seed. That is, what kind of microbes can we put into these ecosystems to try to restore eubiosis and bring benefit to the host. But if you think about it, if you have an inflammatory environment, as an example, it's very difficult for commensal microbes to engraft into that population, into that ecosystem. They just are not very fit in that environment. And so what you see in vitro may not be translated to what happens in vivo, and we have to realize that. Now, unfortunately, they're not very good systems to be able to bridge that gap at the moment, and this is something that I think we need to think about and maybe come up with some potential approaches. The other part of this is that very little attention is paid to the soil part, the ecosystem. And if you don't change that, you're never going to get back the population or have it do what you want it to do in an ecosystem where the organisms are either not fit or they function in a very different way. Now, there are a few things that are developing. The development of hydrogels, nanoparticles that can be targeted to specific regions of the bow. But by and large, this is a fairly neglected area, which I think we have to consider in developing LBPs. So these are sort of my ending talking points for considerations for future LBPs. First of all, I think for human subjects, they have to be from human sources. There is a priority effect. That is that indigenous microbiota, even a dysbiosis, will be fairly stable and very difficult to break when you try to superimpose an FMT. You can get around that by pretreatment with antibiotics, and that has been successful in increasing engraftment and sustainability. The other question, are all gut microbes equal? And the answer to that, I think, is clear. No, they're not equal. They're very, very different, even at the strain level. Engraftment, and this is something that I think we'll talk about during this conference, but I think that to get sustainability, I believe you need engraftment. We need better quality control and standards, particularly for undefined but also defined FMTs. Does one size fit all? I doubt it. Everybody is different and our needs are different. And we may have to start thinking about FMT in a very personalized way. But how do we do that? I mean, the metrics that we have for determining health, individual health, or health of an individual's gut microbiome are just not there. And, but we, I think, can develop certain functional measures that might provide more precision. And then we haven't really thought much about unrecognized short-term and long-term consequences of live biotherapeutic products. I mean, we look at symptoms and we look at whether or not it's safe, but can we do more? So this is the final slide that I have, and the take-home message is this. Yes, I think there is a bright future for live biotherapeutics and for microbiome-based interventions. However, I think that we have to be cautious. We have to do the diligence in order to do good science, to understand and bring in ecological principles that will allow us to make better products that are more precision. We have to design it in a way that is not only effective and sustainable for specific diseases or states of health, but also for the individual. So with that, I think I will end, and hopefully I have a few minutes for questions.
Yes, we have time for a few questions. Is there questions in the audience?
Congratulations, excellent opening. Would be interesting to use antibiotics to clean the bowel and then use fecal transplantation, for instance?
Yes, I think so. That's already been done in several clinical trials. For example, in patients with ulcerative colitis, the Australian group has shown that they can increase the remission rate by applying multiple treatments of FMT preceded with a clearance with antibiotics. Balfour.
Yes, Gary, that was absolutely outstanding. And I think you gave an extremely broad overview. I think your idea of what are the metrics that we can determine a healthy gut is a key takeaway point for me, anyway. And I wonder if you could comment on perhaps looking at functional metrics rather than name of bacteria, what is its function, perhaps with a readout of metabolomics or other indicators of how those bacteria actually convey their protective effects.
Well, that's why I liked your paper, because that's exactly
that
what
was not
done.
a personal plug, but thank you.
But I'm with you, Balfour. I think that function is much more important than taxa-calling, because if you're 16S brings you at best down to a genus level. Well, microbes are so different at the genus level. It's impossible to ascribe any type of function to them. Even if you bring it down to a species and even strain level, strains will differ in both their genome as well as functional properties. So the ultimate goal is to find functional metrics that will allow us to and guide us in developing these types of interventions.
Yeah. And I think another is provide the correct substrate with dietary and perhaps environmental factors.
Thank you. Philippe Marteau?
Yes, so you suggest to treat first with antibiotics and then another treatment. How do you select the antibiotics to decrease the colonization resistance? And do you consider there could be some risk in giving antibiotics to the diseased microbiota?
Yes, great question. I don't have an answer, except that I think what you are, the end goal here is to try to break stability and the resilience of the indigenous microbiota. Now, in C. diff, that's not a problem because they're already gone. But in somebody that may have either a eubiosis or a dysbiosis, it's hard to insert new microbes into that stable population. So you have to sort of perturb it. And there may be other ways besides antibiotics, but antibiotics definitely can improve the engraftment and sustainability of a FMT. So there are other ways. I think that Balfour mentioned some. We could, change the diet in some way that favors the fitness as well as engraftment of certain microbial populations that we want to put in. And so there are other ways to do it.
So maybe the last question.
Hello. I have a question. I really like very much your presentation. In your opinion, to what extent the microbial pangenome should be considered for the effectiveness of the LBTs?
I'm sorry, I couldn't hear the last part of your question. Could you repeat it?
Yes, the microbial pangenomes. The collection of all the genes that are part of the microbial inheritance, all the genes, the pangenomes.
Yes. So is it that what kinds of, I call that metagenomes, are necessary for, let's say, repairing or maintaining a population of, let's say, dysbiosis? Is that the question?
No, what I was trying to say is that I learned that all microbia have extra genes, and those are considered the pangenome for each species. So you mentioned something very interesting that there is not one size that fits everybody. But my point for an expert like you is, to what extent you consider in your experience that we should look at also to the pangenomes of the gut microbiota, for instance? Thank you.
Yeah, I think that we can certainly learn a great deal from the pangenome and looking at accessory versus core genes that are part of that metagenome. In many ways, that can give you a great deal of functional information and also explain why certain types of microbial strains may differ from others. And I think that that may be part of the toolbox that we use in the future to determine which microbes are not only best individually, but also work well together. Hopefully I answered your question.
Thank you very much, Jean.
Thank you.
Okay, we continue with the program, and we are now in plenary session 1, and the topic is next generation probiotics. And it's my great pleasure to invite the first speaker, Michael Charles. Michael is a great expert in innate and adaptive immune response, IBD, and cancer, and also on the role of microbiota in these disorders. So his talk is going to be dealing with the commensal clostridialis strains mediate effective anti-cancer immune response against solid tumors. Please, Michael.
So, thank you very much for the kind introduction, and thank you very much for the organizers for the kind invitation and the opportunity to present you our data on how we could potentially use Clostridialis bacteria as an effective anti-cancer therapy. So the first question we have is, how actually can bacteria contribute or modulate cancer growth? And Laura Zitvogel has 2016 published a nice summary where you can see that bacteria on the one hand can modulate either directly or indirectly the cancer cells themselves, either by promoting proliferation or affecting their apoptosis. And of course, the bacteria, the microbiome, is able to affect the immune cells in our body. So either they can activate specific immune cells, they can inhibit specific immune cells, and vice versa. If the immune cell, the anti-cancer immunity is affected in our body, it is obvious that cancer cells can either grow better or worse. So in melanoma patients, it has been nicely shown that there are specific bacteria that are clearly associated with either response or non-response to those checkpoint inhibitor therapies. And the bacterium that pops up all the time is actually Fecalibacterium prausnitzii, which is indeed a Clostridialis bacterium. And in 2018, the group of Zitvogel has shown when they use fecal microbiota transplantation, the feces from melanoma patients that had nicely responded to checkpoint immunotherapy, they could actually induce a better response in their mice undergoing a melanoma model when they were co-treated with the checkpoint inhibitors. But as we heard previously in the talk from Professor Chang, FMT is a very rough approach, and we actually do not know what is actually the agent causing an effect, because we have simply all bacteria, all molecules that are within the stool. So a maybe more wise approach would be to use specific bacteria. And in 2015, it has already been shown that specific bacteria, in this case Bifidobacterium subspecies, are indeed able to boost checkpoint inhibitor response in mouse models. So the authors first did cancer models. They treated the cancer mouse models together with anti-PD-L1 therapy. They had a look which bacteria were enriched in the stool of the mice that are nicely responded, found this big Bifidobacterium, and then they added the Bifidobacterium plus the anti-PD-L1 therapy and found that the therapeutic effect was much better when they had the bacteria combined with the cancer immunotherapy. And on a molecular level, they found that the combination greatly enhanced anti-CD8 positive T-cell responses in the mice. So in the next step, published in 2019, the authors took actually human stool put the human stool into germ-free mice and had a look in which cell body compartment that this human stool, would induce the biggest CD8 response. They found the colon, and then they assessed which kind of bacteria or which composition of the bacteria would actually be most beneficial and most potent to induce the CD8 response. So they came up with a specific 11 mixed bacteria that also again contains a big number of Clostridiales bacteria, used these 11 mixed bacteria in different cancer mouse models, and then found that the addition of these 11 mixed bacteria was able to boost the checkpoint inhibitor therapy in their cancer mouse models, either if they applied CTLA-4 antibodies or whether they applied anti-PD-1 therapies. So in the next step, and that's the last paper I would like to show you from the background, is the authors did an even further approach. So they treated their mice undergoing cancer models with anti-PD-1 or anti-CTLA-4 therapy, and then analyzed the tumor tissue and analyzed the microbiome and the bacteria that had invaded the tumor tissue. What they found was that particularly in this tumor tissue, specific bifidobacteria were enriched, and then they again performed new mouse cancer models, treated the mice with anti-CTLA-4 checkpoint immunotherapy, added specific bacteria, also those bifidobacteria, and found, interestingly, that this addition of the bifidobacteria, the specific bifido strain, was able to boost the effect of the anti-cancer immunotherapy. But then they went a step further and said, okay, how can the bacteria do this? So they analyzed the serum of the treated mice and found that a specific metabolite called inosine was highly enriched in the serum of the mice that had been treated with this bifidobacterium. And then they did a bunch of very sophisticated experiments and found, in the end, when they blocked the cellular receptor for this inosine, they could completely abolish the effect of the bifidobacteria, clearly suggesting that the effects of the bifidobacterium might be mediated via this inosine metabolite. So, to summarize this first part, can we actually use bacteria as cancer therapy? Well, when we think from mouse models, yes. Fecal microbiota transplantation has nicely been shown to work in mouse models as anti-cancer therapy, and also first data have been published that it might also work as anti-cancer therapy in addition to checkpoint inhibitor therapy in human cancer patients, particularly melanoma patients. What has been shown is in all these studies that the bacteria enhance the efficacy of the immune checkpoint inhibitor therapy, and their early clinical trials are ongoing, for example, using this 11 bacteria mix that I showed you before. And this is a very interesting finding, bacterial metabolites might be also able to enhance the ICI efficacy in our mouse models. But what we were particularly interested in Zurich, and that's the data I would like to show you now, is can we actually use bacteria as a standalone therapy? Can the bacteria actually work if we do not combine them with the checkpoint inhibitors and do their job as an anti-cancer immunotherapy? So for this purpose, we choose a bit different approach and started clearly by the colorectal cancer patients. So in a collaboration with the German Cancer Research Center with a group of Professor Georg Zeller, we actually did a meta-analysis of all published studies where it has been demonstrated how the microbiome composition in the colorectal cancer patients might differ from the microbiome composition in the intestine of healthy people. What you could clearly see is that there is a huge difference between healthy and cancer patients, and we specifically identified specific Clostridiales strains, for example, Rosiburia intestinalis, Eubacterium hallei, or Fecalium prausnitzii, to be highly reduced, up to 5 log-fold reduced in the intestine of the colorectal cancer patients. So we then made a top 10 list of the mostly reduced bacteria and brought those bacteria either as single strains, combinations, 4-strain combinations, into overall 7 different cancer mouse models. So in the first instance, I would like to show you the data from the inflammation-associated colorectal cancer model, which is called DSS-AOM. So we induce a chronic intestinal inflammation by the repetitive addition of the DSS to the mice, inject the AOM, which is a mutagenic substance, and then the mice develop, during the course of this 3 months experiment, colon tumors on the foundation of the intestinal inflammation. So we had either placebo-treated mice, where we orally gavaged sodium chloride. we had mice where we treated Peptostreptococcus stomatis, which is in generally regarded as a tumor-promoting bacteria. And we had a group of mice which we treated with a formix of those bacteria I showed you before, contributing of Rosiburia intestinalis, Fecalibacterium prausnitzii, Eubacterium hallii, and Anerostipes caccae. And what you can clearly see in the endoscopy images, also in the statistical analysis, this CC formix, these 4 bacteria, when we orally gavaged them to the mice, almost completely abolished or prevented any type of tumors in the colon of the mice. And when we then had a look by flow cytometry on the immune cell composition on the intestine in either the tumor tissue or the adjacent non-tumor tissue, we found that the administration of our C-formix reduced the numbers of regulatory T cells or anti-inflammatory macrophages, but critically promoted the levels of natural killer cells and interferon gamma positive, CD8 positive T cells in the colon of the mice, and even more specifically in the tumor tissue of those mice. We then had a look on the microbiome composition after those 3-month treatment period of the mice, and what we found was not only that Rosiburia and Anerostipes were enriched in the tumor tissue and in the intestine of the mice, but we in general found a dramatic shift of the entire mouse microbiome towards Lachnospiraceae and towards Ruminococcae in the intestine of those mice. So the administration of those 4 Clostridiales strains caused a dramatic shift in the intestine, in the intestinal microbiota of our mouse, towards a microbiome that would generally be regarded as a much more good one than it was before. So in the next model, we injected mouse cancer cells, the MT38 tumor cells, subcutaneously into the mice because we wanted to get rid of the impact of the inflammation, and there we injected the cancer cells subcutaneously, applied again either sodium chloride orally to the mice or our formix bacteria orally to the mice, and what you can see is that also in this tumor model, the bacteria had a clearly beneficial effect with respect to the cancer treatment. The bacteria caused a massive infiltration of CD8 positive T cells into the tumor tissue, and interestingly, what the bacteria also did was they downregulated the levels of checkpoint molecules such as TIM3, CTLA4, or PD-1 on a CD8 positive T cell surface. So indeed, they actually acted somehow as in cancer immunotherapy on their own. And then, consequently, in the next step, we wanted to compare the efficacy of our bacteria compared to the anti-PD-1 immunotherapy in our MC38 model. So we injected the cancer cells again into the mice, and at day 6 we started either treatment with a bacteria, with an anti-PD-1, or with a combination. And surprisingly, or interestingly, what we found was that the bacteria were actually at least, if not even better, than the nivolumab treatment in our therapy. And if we had the combination treatment, we also did not have an even further anti-tumor effect compared to the bacteria treatment alone. So then we applied single strain bacteria of our CC formix in our mouse models. So we applied again sodium chloride, our 4 bacteria mix, and each of the single strains separately to the mice. And what we then surprisingly saw was that specific strains, for example, Rosiburia intestinalis and also Fecalibacterium prausnitzii, were even more effective than this 4 bacteria mix when they were applied as the single strains. And we then confirmed those data in a breast cancer model, in lung cancer models, in different mouse facilities all across Zurich, and also in a B16 melanoma model. And here again, we also could see that particularly the application of Rosiburia intestinalis as applied as a single strain was at least equally efficient as the combination of these 4 bacteria mix. So what can now be the conclusion? Well, bacteria might actually be useful as a cancer therapy. The microbiome, and this has nicely been shown in recent years, modulates anti-tumor immune responses in the human body. Bacteria can serve as a predictive biomarker for therapy response. This is particularly shown in the melanoma studies, and they can also enhance, at least the mouse model, somehow immune checkpoint inhibitor efficacy. So now ongoing early clinical trials have actually to prove the efficacy of these bacteria as cancer therapy in humans. And if our own approach would turn out to be also useful in human setting, it would be somehow novel, because we actually would use bacteria not in combination with the cancer immunotherapies, but as standalone therapies to treat cancer in human patients. And in the end, I would like to thank all the people from my lab that contributed to the research, all the collaborators and all the funding institutions. And thank you very much for your attention.
Thank you very much, Michael. That was really fascinating. I think that we have questions from the audience while we wait for people to ask questions. I would like to ask you a couple of questions. First is the relationship between the microbiota and cancer in the colon, colonic cancer. I think that most of the studies look at the fecal microbiota. I was wondering whether is there importance to look at the biofilms and the microenvironment stick to the cancer there, and whether you think that these differences between the fecal microbiota and the resident microbiota is important for cancer progression.
Yeah, I definitely think so. I mean, what is shown that Fusobacterium nucleatum, for example, is highly enriched in the cancer tissue in the cancer patients, and that is shown in other studies, but also in our own research, that there are differences between the fecal microbiota, between the microbiota within the tumor tissue, between the tumor adjacent tissue. So I think it certainly makes sense not only to look at the fecal microbiome, but also what is really going on in the cancer tissue, in the intestine, in the mucosa.
Yeah, and second question I wanted to ask you is, you are looking at single molecules in the metabolome, and this is obviously reasonable, and looking for targets in the tumor cells. I was wondering whether you believe more in a single molecule or in a consortium of molecules that could have an effect on cancer.
That's a very good question that we might hopefully answer in 10 years. I don't know. I think there are data showing that the consortia could be very useful. When you have a look at our own data, I would potentially rather go for single strains. Whether this is true in the human setting, I can answer hopefully in 10 years.
Okay, thanks very much. We have one question, second row.
Yeah. So, Irat Sopani from France. Thank you very much. I really enjoyed, I'm here. I really enjoyed your presentation. Two quick questions. The first one relates to the effect of cross-divisions. Did you have any opportunity to analyze macrophages? You mentioned that macrophages were influenced. Did you see if there was any difference in terms of polarization, M1 versus M2? And the second question, in your experimental models, you used serum in animals, CC4 animals, if I really guessed correctly. I was so surprised by this huge modification in terms of gut microbiota. I was wondering what was your control for such designs?
So the first question, actually, yes, we saw a big change in the macrophage polarization. So when we treated the mice with the bacteria, there was a huge shift from M1 to M2 or vice versa, depending on whether we had Peptostreptococci or the CC4 mix.
And they fitted with this CD8 effects in terms of reducing or increasing tumors.
So the beneficial bacteria clearly increased the numbers of M1 macrophages and decreased the numbers of M2 macrophages.
Got it.
And the second question, what we had as a control group in many of our mouse models were these either Peptostreptococci or Akkermannsia, to have some specificity. And the Peptostreptococci, in general, promoted tumor growth or prevented, for example, the anti-tumor efficacy of the checkpoint inhibitors, and this was somehow the control that not any bacteria we apply to mice would do the same thing with the immune system, because we always had clear differences between Peptostreptococci treatment and CC4 bacteria or the Rosiburia or whatever.
So you didn't use any universal serum just to check if there's what was the background of modification in terms of gut microbiota modification?
So to which mice we normalized the microbiome composition? Or what?
In any subgroups? No. I mean, can we speculate that what is the part of the specific effect compared to the injection of any healthy or universal serum? Can we speculate on that?
And the serum transfer example, for example, we used the serum from healthy mice that had not been treated with the bacteria.
Thank you.
Okay, we have another question here, second row. While we wait for the microphone here, there is another question in the back. Do you already have the microphone?
Yeah, thank you so much. Cornelia Schweinzer, Austria. So thank you for the very impressive talk of using probiotics for the only therapy for cancer patients. That was really impressive. I just have one fast question. As far as I understood from your first part of the presentation, you mentioned inosine as a potential modulating effect for the Clostridialis, either to be able to colonize properly or not. So I was wondering, for the follow-up studies, for the clinical trials, will you also analyze the inosine levels? And maybe did you also analyze them in the mouse studies?
So I mean, what we have shown is that particularly Rosiburia and Anerostipes were clearly colonizing the intestine of the mice. So in, for example, this AOM-DSS experiment, we also analyzed, we had to analyze for the journal where we published the manuscript, also the tumors of the mice and the colon of the mice, and there we found enriched levels, for example, of Rosiburia in the tumor tissue and also in the adjacent colon tissue, in the healthy colon tissue. So I think based on the mouse models, I would say yes, these bacteria somehow to at least some extent engraft also for a longer time in the intestine.
Thank you.
Yes. Yeah, Pierre Dischler, thank you very much. When we skip from the bacteria incubation media to direct injection of the metabolite, there's a question of dose. And it might be that you use very high dose of 3-oxide hydroxide dodecanoic acid as compared to what is produced in vivo. And with a high amount of medium chain fatty acid, you could have some specific metabolic effects, inducing, for instance, the production of ketone bodies and also changing the partition of energy provision to the host and reducing the energy provision to the tumor, and it might be some kind of Warburg effect. So what would you think could be a contribution of some metabolic effect to the finally observed in vivo effect on the tumor, besides the immune stimulation, which is quite clear, of course?
Yeah, so I mean, maybe there are several answers. So what we also did, I mean, at the moment we are developing not that much this specific metabolite, this medium chain fatty acid as a cancer therapy, but we rather would like to focus on specific GBR84 agonists. And what we also did is we did concentration curves in first instance for this medium chain fatty acids, and we thought also if we go to much lower doses, we still have an anti-tumor efficacy, but at some stage, particularly if we apply this metabolite, for example, twice a day or so, we could not further boost it. So at some stage there is a saturation level, that's for sure. On the other hand, we were, to be honest, we were rather focusing on the immune cell outcome, so we did not have that much focus on the metabolic outcome. That's true. Sorry for this.
Any other questions? Yeah.
Hello, Garcia from Mexico. My question is, have you checked if Clostridialis infiltrate into the solid tumor in your model?
And no, we only applied them orally. So we didn't inject them into the tumors or so.
But spontaneously, if you have an evidence, if the bacteria are able to infiltrate the tumor.
So in the colon cancer models that we showed, where the colon tumor is, where the tumor is really in the colon, in the DSS AOM model, actually we found the Rosiburia in the tumor tissue. That's true. In the subcutaneous injection models, we didn't look for this.
Thank you.
Okay, I think that we should move on. Thank you very much.
Thank you.
And it's my great pleasure now to introduce Harry Sokol, the chair of our scientific committee. And he will give a talk on Faecalibacterium prausnitzii as potential LBP to boost immune checkpoint inhibitors efficacy. Harry.
Thank you, Giovanni. So my talk will be very in line with the previous one. And so the idea is to try to convince you that Faecalibacterium progeny is a potential LBP to boost immune checkpoint inhibitor efficacy. So I guess you all know the mechanism of action of immune checkpoint inhibitor, which is to blockade PD-1, PD-L1, or CTLR4 to basically reactivate T-cell response toward cancer cells. And of course, as we know that the microbiome has an important role on immune function, it was quite logical to suspect that it might impact the efficacy of these drugs. And indeed, it has been shown now more than 5 years ago. This is a study by the team of Laurent Zidvogel showing in mice, in a melanoma model, that when you alter the gut microbiome with antibiotic treatment, you decrease the efficacy of immune checkpoint inhibitors. And even in early human study, here is retrospective study mixing many types of different cancer, it was suspected that antibiotic treatment might induce a lower efficacy, as here it was associated with shorter progression-free survival and also overall survival. So very early, the question of specific bacteria came up, and many studies have been exploring this question. And one of the bacteria that popped up was Faecalibacterium progeny. So this is another French study published also in 2017, showing that patients with high level of Faecalibacterium progeny in melanoma had a better response to immune checkpoint inhibitor, in this case anti-CTLR4. These results were also published exactly in the same time by a US team in Science, also in melanoma, this time with PD-1 antibody. And again, the strongest signal in this study was Faecalibacterium, which was also associated with a better response. And when you looked at many of these studies, and here this is a meta-analysis of gut microbiota predicting response to metastatic melanoma, Faecalibacterium progeny was among the strongest signal associated with response. After this study, several other studies also pointed out Faecalibacterium progeny. Here, this is a more recent study in non-small cell lung cancer. So Faecalibacterium progeny was not the only bacteria identified here, but it was among the one associated with a better response. And here again, another study in hepatocellular carcinoma. And in this study, Faecalibacterium was the strongest signal, again, associated with response to cancer. So, of course, next the question is to know whether this bacteria is playing an active role in the response to this type of drug, or if it's a pure biomarker. And actually, as early as 2017, this experiment was performed by the US team by colonizing germ-free mice with the microbiome of patients who responded to immune checkpoint inhibitors or who did not respond to this type of treatment. Then the mice were injected with melanoma cells and finally treated with anti-PD-L1. And the answer to the question is that obviously the microbiome is playing an active role in the process, as mice colonized with the microbiome of responders in blue had indeed a better response to immune checkpoint inhibitor. And interestingly also, in this study, the effect was connected with the level of Faecalibacterium. So what is Faecalibacterium progeny? So this bacteria is really a unique member of the human gut microbiome. It is a strictly anaerobe bacterium belonging to the Firmicutes. It's a member of the phylogenetic core, meaning that it is really present in all healthy human beings. And another important characteristic is that it represents by itself approximately 5% of the normal microbiota. So it's really an important bacteria, also in terms of quantity in our gut. And we and many other teams showed that high level of this bacteria is associated with health, and of course we showed this as early as 2008 in IBD. And we also showed that these bacteria exhibit immunomodulatory effect. So obviously we are working a lot on these bacteria, and seeing all these signals with immune checkpoint inhibitor response, we decided to explore this effect a little bit more. So we started by reanalyzing some data that was published. And this is a study published last year by the team of Laurent Zidvogel, showing that Akkermansia mucinifera, one other very important bacteria in our gut, was associated with a better response to PD-1 blockade in patients with advanced non-small cell lung cancer. And this is basically the result of this study. Patients with Akkermansia had a better response to the drug. But the message is a little bit complex because patients with the highest level of Akkermansia indeed had a worse response. So you need Akkermansia, but not too much, basically. And in this study, no other bacteria were analyzed. So we contacted Laurent Zidvogel and Nicolas Segata, and they agreed very nicely to send us the sequences, so we were able to analyze them ourselves. And we did very simple things. So we separated the population in 3 tertiles according to the abundance of Faecalibacterium progeny. And what you can see clearly here is that the more Faecalibacterium progeny you have, the better is your overall survival. And so, for example, if you take an 18-month time point, you see that there is a delta of more than 20% between patients who have low or high amount of Faecalibacterium progeny. So interestingly, when we looked at the metadata, we were able to find other signals. And notably, we found that patients who were treated with immune checkpoint inhibitors as a first line, actually there was no signal regarding Faecalibacterium progeny, while patients who were more advanced in their therapeutic in their treatment and who were treated with immune checkpoint inhibitors as a second line, the effect was there, and actually it was even stronger with a delta of around 30%. Another interesting result was to compare patients who received antibiotics and patients who did not receive antibiotics. And interestingly, in patients who received antibiotics, we did not see anymore the effect of Faecalibacterium progeny. And actually, here you have to remember that we did not give Faecalibacterium progeny. We are talking about the intrinsic bacteria of the patient. And so the fact that the predictive effect of Faecalibacterium progeny level on the response to treatment is lost suggests that indeed the microbiome might have a causal effect in the response to immune checkpoint inhibitors. So we did a multivariate analysis to take into account potential confounding factors, and also by doing that and incorporating all the classical factors, we found that indeed the level of Faecalibacterium progeny, and notably high level of this bacteria, was associated with a decreased mortality of 40%. Then we continue to reanalyze recently published data, and we came across this study that basically compared the predictive factor of the microbiome in multiple cohorts of patients with advanced melanoma. And the main message of this paper was that they found basically different results from one study to another, suggesting that obviously this is more complicated, that one bacteria fits for everyone. So we got also this result from Nicolas Segata, and we reanalyzed it looking specifically at Faecalibacterium progeny. And when we took all the patients from these 5 cohorts together, this is the signal we found. So there is a weak signal suggesting that high level of this bacteria is indeed associated with protective effect, but as you can see, this is relatively weak and not statistically significant. But again, this is a mix of very heterogeneous population from different countries. So we did a clustering analysis. It's an unsupervised analysis to cluster the population, and we found 2 clusters that was significantly different from each other. And when we looked at the second cluster, which represents 1/3 of the global population of this study, there was absolutely no signal related to Faecalibacterium progeny, while in the other cluster, which represents 2/3 of the global population, we found again this signal related to Faecalibacterium progeny and its protective, potential protective effect. So again, many association studies, some data suggesting causality, but the next question is, can we do something for all these patients with low level of Faecalibacterium progeny to try to improve their response to this type of drug? You probably know these two pilot fecal microbiota transplantation study that were published 2 years ago in patients with refractory melanoma who lost response to immune checkpoint inhibitors. And in these two studies, which were very small and uncontrolled, so many limitations, of course, but still doing FMT was associated with some clinical benefit in 3 out of 10 patients in the first study and 6 out of 15 in the second one. And when you looked at the microbiome data of this study, again, you see some signal with Faecalibacterium progeny, and notably, you see that in the study from Spencer in Science, Faecalibacterium progeny was enriched in post-FMT microbiome of responders. So the question is, can we improve response to immune checkpoint inhibitor with this bacteria? So we took advantage of a strain of Faecalibacterium progeny called EXL01, which is actually currently developed for the treatment of Crohn's disease. So it is possible to use it for clinical trials. So we wanted to evaluate the effect of this bacteria in an in vivo model. So we did a very classical experiment using the sarcoma MCA205 cells. And we use these tumor cells to inject mice that were either treated by antibiotics or not treated by antibiotics. And then mice were treated with anti-PD-L1 and or not with the bacteria EXL01. Here, the antibiotics are given to induce some kind of perturbation of the microbiome. This is not an antibiotic cocktail that will completely wipe out the microbiome, and actually you are going to see it. So here are the results. First, we saw, and this has been published, that of course, anti-PD-L1 is working quite well to reduce the tumor growth, and this is the red curve. And you can see that in orange, the mice who were treated with antibiotics had a less favorable outcome with a not so good response to this drug. This has been published many times. When on top of antibiotics, we gave EXL01, we were able basically to come back to the initial response of immune checkpoint inhibitors. And actually, this is even clearer when you look at survival curve. Here you have in red the anti-PD-L1 treatment group, in black the vehicle group, and again, anti-PD-L1 is working in this model very well. If you give antibiotics, this is the orange curve, you lose a lot of the effect of the anti-PD-L1. And if on top of that you add back EXL01, Faecalibacterium progeny, you are able to recover the normal response to immune checkpoint inhibitors. So we wanted to understand a little bit more what is happening, and so we took biopsies at day 13 in the tumor and did RNA-seq. And I'm just going to give you very simple messages here. First, we just counted the differentially expressed genes. And here this is a comparison between non-treated and treated mice with anti-PD-L1. As you can see, there are almost 4,000 genes that are significantly differentially expressed, and basically when you look at the function, you see increased immune response and decreased cancer cell proliferation. When you look at the effect of antibiotics, so this is antibiotics plus anti-PD-L1 compared to anti-PD-L1 alone, you see again a lot of differentially expressed genes, more than 3,000, and basically you see that cancer cells proliferation associated pathway are increased. If on top of antibiotics you add EXL01, again, you have a lot of differentially expressed genes, more than 4,000, and in terms of functional pathways, you see a decrease in the proliferation of cancer cells. Finally, what is particularly interesting, I think, is what you see when you compare the mice that are treated with anti-PD-L1 alone and the one treated with anti-PD-L1, antibiotics, and EXL01. And in this case, you see that you have a very small number of genes that are differentially expressed, suggesting that basically by bringing these bacteria, you compensate the effect of the alteration of the gut microbiome. You can also see this message by doing PCA analysis. So here in red, each dot represents mice treated by antibiotics, anti-PD-L1 plus EXL01, and in blue, the mice treated with antibiotics and anti-PD-L1 alone. And you can see that in this unsupervised analysis, there is a clear clustering suggesting that there is an effect of EXL01. And when you do the same analysis by comparing mice treated by anti-PD-L1 alone or anti-PD-L1, antibiotics, and EXL01, you see that all the mice are together at the transcriptomic level, suggesting again that you compensate the alteration, the altered response to anti-PD-L1 induced by antibiotics. So, next question, of course, we are always thinking about the microbiome. We are giving some bacteria. Is there any signal in the stool? So we did sequencing analysis on these mice, and I'm just showing you one of the time points, but it's the same for all of them. So here it's for the diversity of the microbiome. So as expected, giving antibiotics to these mice will decrease the diversity of the microbiome, but actually bringing back EXL01, Faecalibacterium progeny, has no effect on the diversity of the microbiome. More interestingly, when we looked at the composition of the microbiome, this is what we saw. So on the left, you have the mice treated with anti-PD-L1 and EXL01, and on the right, the mice not treated with EXL01, and all of them received antibiotics. On top, you have the phylum level and below the family level. So you can see that before treatment, the groups are strictly comparable. And when you go forward into the timeline of the experiment, actually you see nothing. There is no difference at the fecal level when you compare mice who received EXL01 by oral gavage, of course, and mice who did not receive EXL01. So this suggests many things. It suggests one very important aspect is that at least in mice and in this setting, probably you don't need engraftment to have effect with these bacteria in this particular setting. You don't need also to have fecal microbiota modification. However, of course, here we are looking at the stool. So and this has been pointed out by Jean earlier, this is very different from what you have in the small intestine. And indeed, it means that this effect might be related to direct effect on host cell in the small intestine. Of course, this is just hypothesis at this step, and we need to go further to demonstrate it. So then we looked at effect on immune cells, and we did mostly in vitro work for now. We first stimulated T cells from humans, from healthy humans, with EXL01, and these cells were also treated with anti-CD3, CD28 beads to stimulate the TCR, and also with anti-PD-L1, PD-1. So in this context, and again, this is a very specific context, we saw that T cells stimulated by these bacteria will produce gamma interferon with a clear dose effect. And then we did similar experiment with dendritic cells, and in this setting we saw that activation markers were stimulated, such as CD80, for example. PD-L1 was also stimulated, and on the other hand, regulatory markers such as DC sign its expression was decreased. Then we did a mixed lymphocyte reaction assay in presence of nivolumab, so anti-PD-1, and with or without EXL01 at different doses. And what we saw was that EXL01 induced a massive increase in the production of gamma interferon. You see that the production is multiplied by almost 5. And we saw also a slight signal, at least in 2 of the 3 donors we tested regarding the proliferation of T cells. So, in conclusion, the mechanism of the effect of EXL01 strain are still under investigation. It seems that they involved effect on DCs and T cells, potentially regulation of PD-L1 expression. It seems to also to boost T cell function and particularly interferon gamma production. And it's likely not related to change in the fecal microbiome with all the limitations associated with the mouse model. It might be very different in human. And it seems also that the effects are related to some direct stimulation of host cell, probably upstream in the upper GI tract and probably in the small intestine. So the gut microbiota, of course, is involved in efficacy of immune checkpoint inhibitor. I hope I have convinced you that high amount of Faecalibacterium progeny at baseline is associated with better response in multiple types of cancer. Also, the effect might not be the same in all population because of basal microbiome structures that might be different. The EXL01 strain of Faecalibacterium progeny boosts the effect of immune checkpoint inhibitor in vivo, and obviously the next step is to evaluate the effect of these bacteria in clinical trial, and actually this is planned to start hopefully early next year. So I want to thank everyone who were involved in the data I showed, particularly Marius Bredon, who is a bioinformatic postdoc who did all the reanalysis, and also Camille Dan, who were involved in this project, all our other collaborators, and you for your attention.
Thank you very much, Harry. This is very interesting and opens the field to clinical application of these next generation probiotics. And I think that there will be many questions for you. Maybe I can start with a couple of quick questions. The first is whether you can guess or tell us a little bit something about the studies that you're doing on the identification of the metabolites that probably Faecalibacterium progeny produces in terms of efficacy in these models.
So we think, at least in this setting and in mice, that most of the effect is related not really to metabolites that are produced, but to molecules that are present on the membrane, on the bacterial wall itself. Actually, we are working now a lot on this, and we found that the bacterial wall of Faecalibacterium progeny is very specific. And so we think, at least for some of the effect, that it's related to bacterial wall molecules.
So, for example, the effects that you see on the T cells cannot be reproduced with the cultured medium?
Absolutely.
You need the actual bacterium there?
Yes.
Okay, that's very interesting. Yeah, we have many questions. First, here, second row.
Nice presentation. And this is, I think, a very exciting field for probiotics and living organisms to see how it might better increase the efficacy of cancer treatment. So the question, although I think you just answered that, was that when you're thinking about the goal of how we want to give this as a therapy, is that do we want to identify a strain that produces a metabolite that increases the efficacy, or focus on the strain and have it as a production model and then give the purified active metabolite? Again, Harry, should we focus on the metabolite or the whole bacterium?
So if I take my physician hat, I would say I don't care. I just want to treat my patient and do it as fast as possible. So I would say the first step would be to maybe use the low-hanging fruits, and if we have something to propose to our patient, probably use it. And if it's a whole bacteria, why not? If I take my scientist hat, then I say, no, we should use a very purified molecule, act on a very specific target, and hopefully this is what we will achieve at one point. But actually, it's exactly what Jean said. We have to walk before we can run. And so probably we have to use this type of strategy, which are maybe not 100% satisfactory on a scientific point of view, but might be efficient for our patients.
Here, first row. Maybe there we have the microphone. Can we bring a microphone here, first row?
Maybe I can start, Harry?
Yes.
Thank you. Yeah, thank you for your presentation. I think it was really mind-blowing. So I think your ability to prevent the antibiotic-induced detrimental effect is really clear. I'm just curious if you have tried to do fecal microbiota transplantation from non-responder patients and see if you can convert the responsiveness?
Not yet.
Okay. Okay. But this is planned, I guess?
Yes.
Okay, thank you.
Do we have a microphone here for the first row?
Thank you. That was terrific, and thanks for updating us on all of the Prosnitzi journey. When you showed the non-shift in the microbiome, my immediate thought was, do you need live bacteria to create this effect, particularly when you showed your in vitro analysis?
So this is a very good question. We don't know. The thing is that in mice, we cannot protect the bacteria. We give it into the stomach, and this bacteria is very sensitive. So it is very likely that the metabolic activity of the bacteria after it is given to the mice is very low. So at least in this setting, it seems that we don't need to have, active, metabolically active bacteria to have this effect. In human, it might be different because we are able to protect the bacteria, to keep it active metabolically, and so we might actually have even better effect, notably because, of course, Faecalibacterium prosnitzi produce short-chain fatty acids, and there are also data suggesting that butyrate is boosting actually the function of T cells in terms of interferon gamma production, and we might also see some kind of, I don't know if it will be engraftment, but at least maybe some kind of persistence of the bacteria in the gut.
Okay, we have time for a couple of questions. I see many hands.
Okay, Harry. What's the difference between a high concentration of F-Pro and a low one? Do you see several populations or one population with more or less? And the second question is, can we use fibers to boost the endogenous levels of F-Pro?
Yes. So for the first question, what we did is very simple. We just separated the population in tertile. Actually, we did not look at what you are suggested, and we are going to look at it, whether to know whether it is really a binomial distribution or more a continuum. We are going to look at it. And regarding the fibers, actually, we are going to have a very nice talk about it by our colleagues from Canada later.
Yes, Harry, here?
Yeah.
Congratulations. Actually, you're a clinician, you're a scientist, and you're also a businessman. So my question is, whatever works, probably if you observe that the wall membranes, the wall cells are enough to act, then you will skip the LBP regulation and you come back to the postbiotic regulation, which is not so clear. So maybe it's easier for the time to market to use these kind of cell walls because you can demonstrate more easily and have some standardization of the product easier than with live bioproducts. What do you think about this?
This is a good point. Actually, using non-viable bacteria has other drawbacks. But yes, this is a very good question. For the moment, the good thing is that we have a bacteria, a live bacteria, that we can try in clinical trial, at least as proof of concept. Then, if we have a good effect, of course, we can think of maybe simpler type of administration, of course. Yeah.
We have time for one last question here, Balfour.
Fantastic work. A quick question would be if the membrane is important. Do you think it's working through antigenic activation, or do you feel that it might be related to TLR activation or other immune pathways, innate pathways.
We don't know yet, but apparently it's not antigenic.
Okay, we should move on. Thank you very much, Harry. And we move to the next presentation. The speaker is from Calgary, Canada, and she's Marie-Claire Arrieta, and she's an expert in dealing with very young infants, and her talk is dealing with the probiotic to accelerate gut microbiome maturation in extremely preterm infants. Please.
Thank you, Giovanni, for that introduction, and thank you for the invitation to be here. I'm very excited to be in Prague. So changing gears a bit from cancer to probiotics use in other patient population, and these are extremely premature infants. So our lab, these are my disclosures. None of them actually impact what I'm going to say. Our lab is really interested in what happens to the microbiome early in life. We start with 0 microbes in a healthy pregnancy, but quickly that changes, and this community starts getting colonized by an increasing number of microbes, and in the span of a few weeks, there's hundreds of species in the early life microbiome. However, this community undergoes a process of ecological fluctuation, mainly during that first year of life, but continuously up to 3 to 4 years. And what's interesting is that this is happening in parallel with the very drastic physiological changes that occur across all body systems in the infant. And in many cases, these are just 2 parallel processes, that microbiome changes and physiological changes. But also, in many cases, there's crosstalk between the fluctuations in the microbiome early in life and what's happened physiologically, and that's what we're interested in. Now, another thing that I find fascinating is that when we looked at the patterns of succession of the early life microbiome, these are constant across populations. And this is an integrative analysis of 14 shotgun metagenomic studies of the early microbiome up until 48 months of life. And this was done by Corpella and DeVos. And it's interesting to see that those patterns exist, because that tells us that they're likely biologically determined. And we're learning a bit more of what that means and the different processes that are important in that initial process of succession. We also know, as you can see here in red and in blue, that this ecosystem, because it's a young ecosystem, is less resilient and it's more susceptible to perturbations. And that the important key taxa of the early life microbiome can be severely impacted, for example, by the use of antibiotics, which you can see in blue and in the top right, with bifidobacteria, which is known now to be a very important keystone taxa of the early life microbiome, and its abundance can be severely depleted by the use of early life antibiotics. And as we understand, that can have an impact on host physiology as well. However, there's a subgroup of infants, and it's 8 to 10% worldwide, that are born very differently than infants at term. Premature infants are quite different, of course, in terms of their physiology. They're born too soon. They have, the more premature that they get, they can have severe immaturity across many systems, including the respiratory system and the GI system. These are infants that spend many weeks and months in an incubator. They're exposed to many pharmacological treatments that term infants don't have to worry about. Their nutrition is very different as well. It can be parenteral for a few weeks, and combined factors that include prematurity to some of these exposures render them at risk of many very critically dangerous diseases, some of them acute, like neonatal sepsis or necrotizing enterocolitis, but some of them long-term. And some of these disorders, we understand the microbiome can contribute to, at least in the term infant population, but we still have a lot to understand and how the changes in the microbiome and premature infants contribute to this. And this is important because their microbiome is drastically different. So this is again work by Corpella and DeVos. And what they did is that they compared the evolution of the microbiome in 3 groups of infant. On the left, labeled as EP, these are extremely premature infants that were born as early as 25 weeks gestation, but extreme prematurity is everyone born under 29 weeks. The middle group were less premature, but still premature, so the very premature infants born as early as 29 weeks, and then on the right, the control group, healthy term-born infants. And as you can see, there's a very drastic difference, but this matters not only because they're different, but because of the nature of those differences. One of the missing taxa initially in extremely premature infants that you see in red are bifidobacteria, and there's also an overabundance of staphylococci and enterobacteriaceae, and these are some of the agents that are known to be the main culprits of neonatal infections or NICU infections. So, and on top of that, it's been shown that this biotic premature microbiome causally contributes to something like NEC or neonatal sepsis. So because of that, they have been the subject of a lot of probiotic approaches to remediate some of this dysbiosis. However, the use of probiotic really changes in NICU settings around the world, and there's a group of infants for which there's not enough evidence, and this is the extremely premature infants, those on the very left, and the reason for that is that, of course, they're very critical patients, and neonatologists are very cautious as to the type of treatments and interventions that they get. So, because of this, we established a randomized clinical trial at the Foothills Medical Center, adjacent to the University of Calgary, in collaborations with 2 neonatologists, Dr. Al-Shaikh and Dr. Emin, in which we wanted to look at the microbiome of extremely premature infants that were intervened with probiotics. And to repeat, this is not the norm, at least in Canada and many other NICUs. This population of infants do not normally receive probiotics. And the way that the intervention was established was starting at the 2nd week of life. Babies were given daily probiotics, and we organized the sampling events, which you can see by colors there. So we first had an initial pre-intervention sampling event, then we had 2 additional ones in light blue and in purple. Then the intervention stopped about 2 weeks before these babies became term equivalent age, which is about 2 weeks before they go home. We had the first washout sampling event then, and then 6 months after, so 6 months corrected, we had another view of their microbiome. Looking at the variables, comparing the control and the intervention group, there really weren't any differences in terms of some of the clinical variables and other variables that are important in the context of probiotic and microbiome. And this is the preparation. In Canada, is known as Flora Baby. This is a multi-strain probiotic. There are 4 bifidobacterial strains, including Breve, Bifidum, Infantis, and Longum. And there's lactic acid Bacillus rhamnosus as well. And as I said before, this was given daily. So one of the first things that we're really interested in, and going back to some of the things that have been said today about engrafting, we really wanted to know if some of these strains were engrafting or not. And for that, we used the genomes that are available for these 5 strains and designed primers specific for each one of these strains. And this is the work of 2 former members of my lab, Dr. Samara and Dr. Mosavi. And they carried out absolute quantification of each one of these strains. And what we found was quite interesting. There were some strains like L. rhamnosus on the left, where when the probiotic intervention was given in times 2 and 3, there was an elevated concentration for them, but then the intervention stopped right after time 3. And then the concentration goes down, and that likely tell you that this is not a probiotic strain that is successful at engrafting, at least in this particular patient population. But then in contrast, we look at B. brevi, which was the fittest of the strains that we found, and not only did it increase during the probiotic intervention, but it continued up until 6 months corrected age, so many, many months after these were given. And to me, what was even more interesting was looking at the control group in gray, and at around time point 4, which is the term equivalent age, these babies start acquiring these strains as well, potentially telling us that they are indigenous strain that can disperse eventually in this patient population. When looking at the changes now to the overall microbiome, this is done by 16S, control on top and probiotics on the bottom. The main change that you can see there is an increase on the bifidobacterium in yellow, which is not surprising because these are the majority of the strains that were given. Again, what's interesting is that they continue to be increased after stopping the intervention, and also in the control group, they start being acquired at around term equivalent age. Now, we decided to take a more unsupervised approach to analyzing the microbiome. So instead of comparing the microbiome by a specific variable, like treatment, we decided to do community typing analysis. And we think this is an important approach, particularly for early life microbiomes, because there's a lot of inter-individual variability, especially very, very early in life. And this is a method used commonly in ecology for this type of thing. And you just cluster the samples based on community composition. And this analysis yielded 4 community types that we labeled originally C1 to C4. Now, when we looked at the distribution of each one of these community types across time points and comparing controls versus intervened babies, we saw that they start very similar, but quickly you could tell that the C4, this turquoise community type, it starts appearing earlier in the probiotic group. And as you can probably imagine, the composition of the C4 community is mainly explained by a high abundance of bifidobacterium. But what were really interesting to know how that compared to the healthy term microbiome. And we partnered with good collaborator Dan Knights and his student Susie Hoops. And they have this really nice cohort, and they sent us samples and data from a group of healthy term babies. They're born healthy, but also they were exclusively breastfed, and they were born vaginally, so what you would call the gold standard in early life microbiome. And what you see here is a comparison of the beta diversity, or the microbiome composition of each one of those community types, now compared to what the healthy term microbiome would be. And the only cluster that approximates that is that C4 cluster. And I think this started providing us evidence that the C4 cluster, this intervene cluster, approximates or matures faster than the other ones. Now, one of the things that we looked at, and it's part of the paper that it's already published, was that, of course, this wasn't the case for every infant. And when we looked at infants individually, some of them went from C1 directly to C4, some of them had a more staged evolution between clusters, some of them stayed at C2, and we're really interested in understanding that. And for that, we decided to measure the probability of transition between clusters, which is what I'm showing here. So a whole bunch of numbers, but basically what you see, what those numbers are telling you are exactly that, the probability of going from one cluster to the next, and that's shown with the arrow. So what we found through this analysis was that in the probiotic groups, the transition from any cluster into the C4 or the mature cluster was a lot higher, was significantly higher. But what was more interesting even was that in both the control and in the probiotic groups, the probability of C4, the mature cluster, to remain as C4 was the highest. And this provided evidence that this may be a more stable community, that once the bifidobacteria come in, this transition into an ecosystem that prefers to stay there for whatever mechanism or reason. This is added evidence that this may be the case. This is network analysis, which looks at co-occurrence of different taxa, and we did that for each one of the clusters, and you can see in comparison to C1, which is the most immature cluster, there's only a few taxa depicted as being significantly interconnected with others, and this is in comparison with C4. This is a more complex community, more interconnected community, also potentially suggestive of community stability as well. Now I'm showing this complicated model because of what it tells you, and let's see with a pointer. Doesn't really work. I'll walk you through it. So this is a structural equation model. And what this modeling does is that it allows us to provide directionality or inferred cause and effect between different variables in a study. And for this, our outcome was the microbiome at T1, T2, T3, and T4. And we use a priori knowledge to hypothesize which variables may be impacting the microbiome. And then what the model does is tell us whether there is a significant relationship and the directionality of that effect. And what we found was that mode of birth, which we show as C/S, was significantly influencing the community at the first time point. But then what we found later was that breast milk consumption and the probiotic were also significantly influencing the community at the time point 2, which is when probiotics really started. But what was most interesting was that the effect of probiotics significantly influencing the community is not significant at T3 and at T4. What we see is that the community itself at T4 influences the subsequent communities. And this is important because this is evidence for this process known as historical contingency. We heard about it earlier, priority effects. Those members of the microbiome that established early on have an important influence in what happens later. And this model provides us further evidence for that. We also found that the changes on the microbiome were not just taxonomical, but functional in nature as well by looking at metabolomics of these samples. It was interesting to see that the metabolome changes with time between T1 and T5, but also that this is influenced by the probiotic. So on the right, you're seeing that those changes in the overall metabolome in the control group, these occur in a more staged fashion. But in the probiotic groups, you have that dark blue hill of the first time point, and once you intervene with the probiotic, that transitions faster into a more mature metabolome. And these are some of the examples now in the context of microbiome maturity of some of the important metabolites that are changing when the microbiome matures. For example, taurine and cholate, which are important building blocks of primary bile acids, which are very important in this population so that they can metabolize the fats in breast milk. They were increased in the microbiome mature samples. And then reciprocally, oleic acid, which is the fatty acid that is most abundant in breast milk, was decreased. So we're thinking that there's a favorable signs of acquiring a more mature metabolome in this patient population. In the lab, we're really interested in looking at the fungi. A lot can be gained from looking at inter-kingdom interactions between bacteria and fungi. And we found that, yes, we're giving a probiotic of only bacterial species, but we found a pretty significant change in the fungi too, and this is by ITS2. The probiotics had a very drastic decrease in the relative abundance of Candida species. And this matters because Candida is one of those top 5 culprits of sepsis in these babies. We inferred their immune function by looking at cytokines in stool. Unfortunately, we're not allowed to take samples from these very premature babies, but we see an overall decrease in pro-inflammatory cytokines during the time that these babies are in the NICU when they're intervened with probiotics, including calprotectin and other ones. And in a separate study now looking at outcomes, what our colleagues did was that they look at many of the different outcomes of clinical importance, and they found that in the intervention group, these babies were less likely to be given hydrolyzed formula, which is normally indicated when there are intolerance to milk, both cow and human milk, which potentially means that there's also clinical benefits to their use as well. But finally, from my perspective as a microbiologist, I think the ecological analysis done here provides really important evidence of what can happen when the right strains are given at the right time, which we heard about before. Bifidobacterium are human, indigenous, certain strains are of the early life human microbiome, and when given early, not only can they engraft, but they can really engineer the ecosystem around them by increasing biodiversity, allowing other species to come in, and providing more stability to that ecosystem. And we think, although we're continuing to study these infants, that this comes with health benefits as well. And finally, I agree with Dr. Chang that a lot can be gained by looking at the phenomena that we commonly observe in microbiome science under the lens of ecology. Ecologically, our microbiomes can be viewed as a meta-community in which our microbiomes are linked by this process known as dispersal. And this important ecological mechanism is what explains microbes going from person to person, and importantly, vertical transmission too. And our results, for example, our quantification at the strain level, what it's showing is that the control infants are unable to acquire these strains until much later, or they do so in insufficient numbers. And this may be because the NICU setting creates a barrier for dispersal because of its settings and the need for them to be. And in this context, the right probiotic can constitute a bit of an ecological restoration approach to break that barrier in a way towards a desired composition of the microbiome in cases where it cannot naturally occur. And with that, I'd like to thank my group in Calgary, different collaborators, and of course financial sources, and the people in bolded letters were the ones that contributed to the data that I presented here. Thank you.
Thank you very much, Marie-Claire. We have time for questions. Yes?
Thanks for the fantastic and inspiring presentation. In your intervention in preterm babies with probiotic mix, you show that Bifidobacterium breve strain colonized and established durably in the treatment group, but also in the control group, so that relates to the dispersal you were mentioning just before. Do you suspect in this case that they could be transferred by the personnel of the hospital?
Yes, I mean, these are, and all NICUs are different, but these are NICUs where, of course, each baby is in an incubator, but there's opportunities for these microorganisms to cross-contaminate, if you may, like any other microbe. But I think that in this particular case, they don't do it, the baby can only acquire them in sufficient numbers until later on, for reasons that I don't understand quite well. What is it about that early phase in extremely premature infants that prevent them from acquiring those microorganisms that are present in the unit?
That's fascinating. We have results from studies in France that show that actually you can point out where the baby was born, just on the basis of the colonization in early life. So maybe that's important to consider for the future. And you show the clusters, very, very interesting aspects. Could you connect this with what we've called enterotypes for the adults?
Yes, although, I mean, it's a very similar approach, right? It is community typing at the end. The enterotype concept is established in the adult microbiome, so very different composition, and is, of course, mainly based on the most abundant species on each one of those. Those, at least, have not really bloomed to that level yet, so they wouldn't equate to what we know as enterotypes, but the method is very similar.
Okay, thank you.
You're welcome.
We have a question there in the back, and then here.
Hello, Garcia from Mexico. Very nice results on using the probiotics in these very early preterm children. I just wonder, what do you think might be the result if you use milk from a human milk bank?
Yes, good point that I didn't mention. In our setting, the majority of these infants are supplemented with banked, not mother's own, but banked human milk, usually a combination, and it's very individualized depending on the growth need of each individual infant, right? But the majority of them start with human milk, combination of moms, if she can produce enough or at all, and banked milk. And also, eventually, they also have to get fortifiers and formula because they need to grow. That's the most important thing clinically. But that changes depending on the NICU. There's a lot of NICU settings where milk bank is not even present. There's a lot of really good evidence really establishing this very close connection between human milk and the ability of bifidobacteria to flourish in the infant gut. So I believe that it needs to be there, and that without those important prebiotic substrates, bifidobacteria is going to have a hard time in establishing itself there.
Okay, here.
Terrific presentation. One of the things that you pointed out early on is developmentally these infants are not meant to have food exposure to their gut or microbiome in their gut. Were you able to demonstrate any difference in what we think of as pathobionts that have been suggested to predispose to some of the things that you look for, like necrotizing enterocolitis?
Yeah, we did. I didn't show it. Others have shown it too. In both extreme and less extreme premature infants, that probiotics, particularly the ones including bifidobacteria, are quite good at decreasing the abundance of Klebsiella, E. coli, Shigella, as well as staph and others. Yeah, and we found the same.
I would encourage continuing to look at this response that you saw in loss of requirement for hydrolyzed formulas as a potential connectivity between prematurity, diet changes, hospital stays, and predisposition to these early food intolerances.
Yeah, I agree. This is part of a larger study. I didn't talk about it today. It's called the Bloom study that we established in Calgary. We have almost 250 infants now, and funding to look at them up until age 3, really interested in how the microbiome is linked to immune development and other parts of development.
Thank you.
I think there is first one question there, and then here, Eugene.
Maybe I missed something. I saw the definition of the probiotics you used as species, but not at the strain level. Do you think that the strain level is important for the effect?
Yes, I do. Absolutely. I think probiotic research should always label the strain, because strains can be very different, particularly in the genus Bifidobacterium. So I did mention we use 4 strains of Bifidobacterium. I put it in the chart. I can't remember their strain designation, but it's there. It's in the paper. And yes, always just use strain designation for probiotic studies.
I really enjoyed your talk. I have a question. You know, the use of antibiotics, particularly in premature infants and NEC, is common. And in light of what you just showed, should we revisit that? I mean, maybe this isn't the best practice in that it may impair the proper succession of microbes to maturity.
Potentially, and I do like where your question is inferring. I mean, it could potentially prevent dysbiosis, but this should also be looked at under the light of prevention of infection, which is very real in that setting as well, and it's what's indicated the use of antibiotics. I think that what may need to change is the amount of antibiotics, at least in our setting, and it changes setting to setting. Probiotics are given as part of the day one pharmacological regime to these infants. They go into the unit and they don't have a real indication for antibiotic use except for their extreme prematurity. So I think that potentially a more balanced and definitely more personalized way of treating them. One of the things that to me would be really important is having more live monitoring of that infant's microbiome, whether it's on pathobiont abundance or the abundance of bifidobacterium, and that way inform some of the clinical decisions a bit better.
You know, in the adult population, we have often patients who undergo stem cell or bone marrow transplantation, and then they are prone to these multi-drug resistant microbes. But Eric Paymer has shown that, you know, if you introduce either FMT or defined microbiota, that you can actually suppress those pathogens and restore eubiosis with better clinical outcomes.
Without the need of antibiotics. Interesting, yeah.
We have time for the last question there.
Fantastic work, Mario Leclerc from Penelon. I wanted to ask you about the ethnicity and then going back to this strain specificity. Because Jack Ravel first demonstrated that there's different strains in the vagina of women of different ethnicity and according to the time. So I was wondering if you were able to look at that, if you had enough power to look at that in your data.
Unfortunately not. This is what we're doing now with the Bloom study. But yes, at a study of 60 infants, we could not look at that factor. Thank you.
Okay, thank you very much. This brings us to the end of plenary session 1. We have a half-hour break now, and then we have to be back in the